Hey everyone, welcome back to this online lecture video series. These are college level lectures for STEM students. We're covering part 2 of cell communication. If you haven't already, have a look at part 1 and make sure that you're comfortable with the content covered there before we move on. This lecture is for you if you missed your lecture, didn't quite understand it, or need a review before an exam. In this video, we are covering topics 4 through 7. Topics 1 through 3 were discussed in part 1 of this lecture. We'll begin with transduction, cover cascade pathways, phosphorylation, and second messengers, then move forward to the cell responses, both nuclear and cytoplasmic, including apoptosis. Peep down in the description box for timestamps if you wish to skip ahead to a particular topic. Here are the goals for this lecture. You'll be able to describe in detail transduction pathways, the role of phosphorylation and small ions, be able to evaluate the role of second messengers in signal transmission, describe response and how it leads to regulation of transcription in the nucleus or cytoplasmic activities, and describe apoptosis and how it integrates multiple cell signaling pathways. Before we dive in, let's make sure that we're all on the same page. Here's a little schematic I put together for this chapter. In the last lecture, we covered the reception box. In this lecture, we'll complete transduction and response. Flowcharts are perfect for simplifying and organizing biological processes, especially complex ones like this. If you'd like a copy to add to your notes, see the description box below. In part 1 of this lecture, we learned that reception is when a target detects a signaling molecule that binds to a receptor protein on the cell surface. Once that occurs, the next phase, transduction, is triggered. Here, the binding of the signaling molecule alters the receptor and initiates a signal transduction pathway, which is a series of specific steps. Often, something is turned on or off, whether that is a gene being expressed or an enzyme being activated or deactivated. So, what is a signal transduction pathway? Once a ligand binds to a receptor, the signal is transmitted through the membrane and into the cytoplasm. Continuation of a signal in this manner is called signal transduction. To accomplish this, cascades of molecular interactions transmit signals from receptors to relay molecules within the cell. To put it simply, it's a multi-step process that can greatly amplify a signal. In addition to amplifying, these pathways give opportunities for coordination and regulation of the cellular response. I want you to note that signal transduction only occurs with cell surface receptors, which cannot interact with most components of the cell, such as DNA. Only internal receptors are able to interact directly with DNA in the nucleus to initiate protein synthesis. Why? Because cell surface receptors are stuck in the membrane. These pathways have a few names, for example, signal cascade and biochemical cascade. So let's start at the top. The binding of a signaling molecule to a cell surface receptor triggers the first step in a chain of molecular interactions. The activated receptor activates a protein within the cell, which activates another, and so on until that protein producing the response is activated. The cascades are usually represented like this, and I'll explain this in more detail in a bit. At each step, the signal is transduced into a different form. Commonly, we observe a shape change in a protein. This shape change is called a conformational change. We saw this in part 1 with ion channels. The binding of its receptor triggers the conformational change, which opens these channels for ions to flow down their concentration gradient. When a ligand binds to its receptor, conformational changes can also affect the receptor's intracellular domain, leading to activation of associated proteins there. In some cases, binding of the ligand causes dimerization of the receptor, which means that two receptors bind to each other to form a stable complex called a dimer. A dimer is a chemical compound formed when two molecules that are often identical join together. The binding of the receptors in this manner enables their intracellular domains to come into close contact and activate each other. And we saw this with the tyrosine kinase receptors in part 1. In my lecture, we will discuss two methods of intracellular signaling. First is phosphorylation and dephosphorylation, which is the transfer of phosphate groups using kinases and phosphatases. The other is second messengers, which are small, non-protein, water-soluble molecules or ions that spread throughout a cell by diffusion. And we'll cover a few different second messengers and their involved pathways as well. Phosphorylation. These modifications are commonly used in cells to regulate protein activity. Phosphate groups will often activate or deactivate proteins. Here, an enzyme called a kinase transfers phosphates from ATP to a protein, and we call this process phosphorylation. Notice in the reaction above, we start with two reactants, protein A and ATP. One of the phosphates of ATP is transferred to protein A, creating ADP, which is adenosine diphosphate. Many relay molecules in signaling transduction pathways are protein kinases, as we will see, and this often creates a phosphorylation cascade. Let's take a moment to look a little deeper into kinases. In this image, you can see the transfer of the phosphate group from ATP to the protein and the subsequent creation of ADP. I'm sure you all know that there are 20 amino acids. 3 of them are shown here: tyrosine, threonine, and serine. Remember that enzymes are very specific for the reactions that they catalyze. There are different types of kinases. Some catalyze the transfer of phosphate groups onto tyrosine residues, and others onto threonine or serine residues. In the image on the right, you can see the amino acid R groups with phosphates attached. Just something to keep in mind. Here's an example of a phosphorylation cascade. We have an original signal ligand bind to its receptor, which activated an internal relay molecule. That molecule activates a kinase, which activates another kinase, all via phosphorylation, which activates another kinase, again via phosphorylation, and so on. A phosphorylation cascade is a string of kinases being phosphorylated and activated. Now, if a cell can put it on, the cell can take it off. And by it, I mean the phosphate group. Here is dephosphorylation. If the cell purposefully activated something to achieve an end goal, it usually will also have to inactivate it once that goal is achieved. Protein phosphatases rapidly remove the phosphates from proteins, which we call dephosphorylation. This phosphorylation and dephosphorylation system acts as a molecular switch, turning activities on and off or up and down as required. Now we move on to the second method of intracellular signaling, second messengers. Many signaling pathways involve second messengers. These are small, non-protein, water-soluble molecules or ions that spread throughout a cell by diffusion. Second messengers participate in pathways initiated by GPCRs, which are G protein coupled receptors, and RTKs, which are receptor tyrosine kinases. Cyclic AMP, which is usually written CAMP, and calcium ions are common second messengers. Inositol phospholipids are lipids that can also be converted into second messengers. Here we have IP3, which is inositol triphosphate. Let's start with CAMP. Cyclic AMP is a small molecule synthesized by the enzyme adenylyl cyclase, or sometimes you'll see adenylate cyclase, from ATP. Notice that two phosphate groups are lost from ATP, creating AMP, adenosine monophosphate. And can you find the cyclic part of the molecule? Adenylyl cyclase is an enzyme anchored in the plasma membrane. And again, it converts ATP to cyclic AMP in response to an extracellular signal. Many signal molecules trigger formation of CAMP. Other components of CAMP pathways are G proteins, G protein coupled receptors, and protein kinases. Look at this pathway. What molecule is activating adenylyl cyclase here? It's a G protein. The signal here was norepinephrine, and it bound to its receptor, which is a G protein coupled receptor. Upon binding, the G protein moves to adenylyl cyclase and activates it. Adenylyl cyclase then synthesizes CAMP, you can also call it CAMP from ATP. The main role of CAMP in cells is to bind to and activate an enzyme called CAMP-dependent kinase, protein kinase A kinase, or simply pKa for protein kinase A. Termination of the signal occurs when an enzyme called phosphodiesterase converts CAMP into AMP. Further regulation of cell metabolism is provided by G protein systems that inhibit adenylyl cyclase. pKa, protein kinase A, regulates many vital metabolic pathways. It phosphorylates serine and threonine residues of its target proteins, activating them in the process. pKa is found in many different types of cells, and the target proteins in each kind of cell are different. Differences give rise to the variation of the responses to CAMP in different cells. In this cell, A lipase is phosphorylated, which triggers breakdown of triglycerides into fatty acids. The cholera bacterium Vibrio cholerae produces a toxin that modifies a G protein so that it is stuck in its active form. And can you think of cellular and organismal consequences for G proteins being stuck in an active form? Again, it will differ based on the cell that we're talking about. As we just discussed, second messengers participate in pathways initiated by GPCRs. They are also involved in RTKs. For a review on these, you can see part 1 of this lecture. Next, we look at inositol phospholipids. These are lipids that can be converted into second messengers. Because these molecules are membrane components, they are located near membrane-bound receptors and can easily interact with them. Phosphatidylinositol, which is PI, is the main phospholipid that plays a role in cellular signaling. Kinases phosphorylate PI to form PI phosphate, which is PIP, and PIP2, which is PI bisphosphate. So there's 2 phosphates on that one. The enzyme phospholipase C, PLC, breaks down PIP2 into IP3, which is inositol triphosphate, and DAG, which is diacylglycerol, both of which serve as second messengers. So here's our second messengers. You can see the cleavage of PIP2 by PLC in this image. Once PIP2 is cleaved, DAG, diacylglycerol, remains in the plasma membrane and activates protein kinase C, PKC, which then phosphorylates serine and threonine residues in its target proteins. On the other hand, IP3 diffuses into the cytoplasm and binds to ligand-gated calcium channels in the ER, the endoplasmic reticulum, to release calcium ions that continues the signal cascade. Take a moment to study this relay pathway. You might even want to pause and draw it out for your notes. This is the activation of a G protein coupled receptor signaling pathway. On the left, the binding of a ligand to the GPCR triggers a conformational change, which activates the alpha G protein, and GDP is replaced by GTP. The activated alpha G protein finds PLC and activates it, leading to the cleavage of PIP2 into DAG and IP3. DAG activates the kinase PKC. IP3 opens ER calcium channels. So let's talk about the last second messenger that's on our list, calcium ions. The calcium ion is a widely used second messenger, even more so than cyclic AMP. The free concentration of calcium ions, which are usually shown as Ca2+, within a cell is very low because ion pumps in the plasma membrane continuously remove it from inside the cell. Ca2+ can function as a second messenger because its concentration in the cytosol is normally much lower than the concentration outside the cell. A small change in the number of calcium ions within the cell thus represents a relatively large percentage change in the total calcium concentration. For signaling purposes, calcium is stored in cytoplasmic vesicles, such as the endoplasmic reticulum, or accessed from outside the cell. When signaling occurs, ligand-gated calcium ion channels allow the higher levels of calcium that are present outside the cell or in intracellular storage compartments of the ER to flow into the cytoplasm, which raises the concentration of cytoplasmic calcium ions. Pathways leading to the release of calcium involve inositol triphosphate, IP3, and diacylglycerol, DAG, as additional second messengers. The response to the increase in calcium varies and depends on the cell type involved. For example, in the beta cells of the pancreas, calcium signaling leads to the release of insulin. And in muscle cells, an increase in calcium leads to muscle contractions. Take a moment to look over this graphic of multiple signaling pathways in the cell. Do you recognize a few of them? Notably, the survival factors binding to RTK in the top left, and the ligands binding to GPCRs next to it. We haven't learned all the pathways showcased here yet, but I hope this complex process of cell communication is beginning to be a little bit clearer for you. I also want you to notice all these cellular components with a K in the name. These are all typically kinases. That's what the K stands for. In a little bit, we will also cover apoptosis, which you can see in the bottom left of the graphic. We are now finished with transduction and just at about halfway through this lecture. Hopefully this schematic is becoming even more simple for you. If you haven't already, take a short break, rest your eyes, rest your mind, walk around, hydrate. This small break will help you focus. And next, we will learn response. So we've finally reached the third aspect of cell signaling, response. During a response, a transduced signal triggers a specific response in a target cell. We cannot talk response without first discussing regulation of response. Remember, a response to a signal may not be simply on or off. The cell is a master of checks and balances. It's constantly taking in stimuli and responding to maintain homeostasis. Homeostasis can be applied to the organismal level and to the cellular level. There are 4 aspects of signal regulation that we will talk about: amplification of the signal and thus the response, specificity of the response, the overall efficiency of response enhanced by scaffolding proteins, and termination of the signal. Number 1, amplification of the signal. Enzyme cascades amplify the cell's response to the signal. At each step of a cascade, the number of activated products can be much greater than in the preceding step. This is a very important concept. One signaling molecule can result in the production or release of many second messengers, which can then activate many other proteins. Note that it is the second messengers which are responsible for amplification of the signal. Amplification occurs as protein kinases, once activated by CAMP, can catalyze many reactions. Some enzymes can catalyze up to 1 million reactions in a second. In this way, a small amount of hormone can trigger the formation of a large amount of cellular product. Here is the phosphorylation cascade that we saw earlier. Number 2, specificity of the response. As I'm certain you all know, each of our cells contains the same DNA, whether it's your muscle cell or an immune cell, it's pretty much identical. Yet, your cells all look and function differently. This is because different kinds of cells have different collections of proteins that are expressed. These different proteins allow cells to detect and respond to different signals. Here we see 2 different receptors which respond to 2 different ligand signals. On that note, the same signal can have different effects in cells with different proteins and pathways. Here we have an adrenaline molecule binding its receptor in 2 different cells. Let's say a muscle cell and a neuron. That adrenaline signal will result in different responses, although those responses are usually a coordinated effort to achieve an end goal. When is adrenaline usually released into the bloodstream? During fight or flight or in a stressful situation. So, this molecule will help the cells to get the body ready in different ways. Pathway branching and crosstalk further help the cell coordinate incoming signals. We saw this in the graphic earlier. Notice how one molecule can affect another pathway. They may activate or inhibit depending on the goal of the end response. Remember, checks and balances. Number 3, efficiency and scaffolding proteins. Scaffolding proteins are large relay proteins to which several other relay proteins are attached. In this image, you can see the large scaffolding protein with 3 kinases attached to it. Scaffolding proteins can increase the signal transduction efficiency by grouping together different proteins involved in the same pathway. In some cases, scaffolding proteins may also help activate some of the relay proteins. And number 4, termination of the signal. Just as important it is to begin a signaling pathway, it's important to be able to turn it off. Often, cancer is the result of not being able to turn signals off. There are 3 inactivation mechanisms here. 1, degrade the signal ligand or remove it. When the concentration of external signaling molecules falls, fewer receptors will be bound. Unbound receptors then revert to an inactive state, and there will be no more transduction. 2. Enzymes reverse cellular modifications from signaling cascades. For example, if a molecule had been activated by kinase phosphorylation, then we need a phosphatase to catalyze dephosphorylation of that molecule. 3. Cyclic AMP is degraded into AMP by the enzyme phosphodiesterase. CAMP is a very common second messenger. If the enzyme phosphodiesterase degrades it, it can no longer trigger a signaling cascade. You may recognize phosphodiesterase as the enzyme that breaks the phosphodiester backbone of DNA and RNA molecules. And now we've finally arrived to the end goal of the signal, the cellular response. Ultimately, a signal transduction pathway leads to regulation of one or more cellular activities. The response may occur in the nucleus or in the cytoplasm, and we can group cellular responses into two categories: responses that regulate transcription or responses that regulate cytoplasmic activities. Many signaling pathways regulate the synthesis of enzymes or other proteins, usually by turning genes on or off in the nucleus. In the first group, regulation of transcription will always occur in the nucleus of eukaryotic cells. Here, transcription factors will be activated, which will regulate synthesis of enzymes or other proteins. If you've never heard of the term transcription factor before, these are sequence-specific DNA binding factors. They're proteins that control the rate of transcription of a gene by binding to a specific DNA sequence and altering RNA polymerase recruitment. In this image, we see a transcription factor binding upstream of a gene start site. The transcription factor will be made of specific amino acids called a DNA binding domain, or DBD, that only binds to a specific portion of the DNA. They bind to enhancer or promoter regions of DNA. Transcription factors can work alone or with other proteins in a complex by activating or repressing the recruitment of RNA polymerase to specific genes. Of course, RNA polymerase is the enzyme that reads and transcribes the DNA into RNA. The function of transcription factors is to regulate, meaning turn on or off, genes to make sure that they are expressed in a desired cell at the right time, in the right amount, throughout the life of the cell and the organism. Other pathways may regulate the activity of proteins rather than their synthesis. For example, opening or closing of an ion channel in the plasma membrane, or change in the activity of a metabolic enzyme. For example, the stimulation of glycogen breakdown by adrenaline for fast energy during times of stress. Another major cellular response is apoptosis, or sometimes people call it apoptosis. Cells that are infected, damaged, or at the end of their functional lives often undergo this programmed cell death. Here, components of the cell are chopped up and packaged into vesicles that are digested by scavenger cells. Apoptosis is a crucial metabolic process for the body. It prevents enzymes from leaking out of a dying cell and damaging neighboring cells. And as a note, apoptosis is distinct from another process called necrosis. Here's a quick summary of the difference between apoptosis and necrosis. Apoptosis is programmed, meaning it's purposeful and targeted. It's generally beneficial for the organism. There are two types of apoptosis, which I'll cover very briefly: intrinsic versus extrinsic. As this is programmed cell death, it is controlled, so no damage occurs to neighboring cells. Components of the cell are again chopped up and packaged into vesicles, which are called apoptotic bodies, and they're digested by scavenger cells, which are typically white blood cells, leukocytes. Conversely, necrosis is traumatic cell death due to acute injury such as frostbite, hypoxia, ischemia. This process is uncontrolled. It's usually detrimental. Here, the cell membrane ruptures and releases contents into the extracellular matrix. And necrosis is also known to trigger an inflammatory response. Here's a quick snapshot at the mechanisms of apoptosis and necrosis. You may not need to know this for your class, but it's still good to compare. Notice that some of the upstream proteins are the same. We have BID, BIM, BAX, and BAC. However, they can induce different outcomes. Notice that apoptosis induces the activation of enzymes called Caspases. We'll see these in a moment. There's also an apoptosome that's formed. For the rest of the lecture, we'll focus on apoptosis. Again, apoptosis is programmed cell death. It's triggered by signals that activate a cascade of so-called suicide proteins in the cells that are destined to die. These signals are aptly named death signals. Often there's a master regulator which acts as a brake and prevents apoptosis. In C. elegans, SED9 is one of these master regulators. Death signals disinhibit that master regulator, allowing the apoptotic signaling cascade to commence. So, what are these cascades of so-called suicide proteins? They'd be proteases and nucleases. Proteases cut up proteins, and nucleases cut up nucleic acids like DNA in the cell. And once again, there are two types of apoptosis: intrinsic and extrinsic. Extrinsic apoptosis is triggered by stress outside the cell. Surrounding cells may be signaling that a particular cell is abnormal, dangerous, or that it has simply moved too far away from the extracellular matrix. We do not want cells to locate to other parts of the body. This is called metastasis and typically occurs to cancerous cells. Extrinsic is receptor-mediated apoptosis. So we have death ligands binding death receptors. As a part of the cascade, caspases 8, 3, and 7 are activated. Other components are TNF alpha, NF kappa B, and PARP. An example of when extrinsic apoptosis is utilized is to destroy bad T cells that target self cells. Intrinsic apoptosis is triggered by stress within the cell. Oncogenes, DNA damage, hypoxia, chromosome rearrangement, to name a few. This is cellular stress-mediated apoptosis. A defining aspect of this pathway is MOMP, which is mitochondrial outer membrane permeability, which results in the release of cytochrome C. You may recognize cytochrome C if you've learned cellular respiration. We'll look at this in a moment. Tumor protein p53 activates pro-apoptotic BCL2 proteins, which results in MOMP, the release of cytochrome C, subsequent caspase activation, and ultimately apoptosis, cell death. An example of when intrinsic apoptosis is utilized is during embryogenesis. Here's an image of a virus-infected cell being induced into programmed cell death by a cytotoxic T cell, which is a T lymphocyte, part of our amazing immune system. On the left, we have the extrinsic pathway activated by the binding of a death factor to the death receptor. This creates a large congregation of proteins called a death-inducing signaling complex, or DISC. This activates caspases and leads to apoptosis. On the right, we have the intrinsic pathway activated by BID, BAX, and BAC. These cause MOMP, mitochondrial outer membrane permeability, resulting in the leaking of cytochrome C into the cytoplasm. An apoptosome is formed, leading to the activation of caspases 9, 3, and 7, and finally, apoptosis. I keep mentioning these caspases, so what are they? Human caspases are a family of 12 cysteine proteases, best known for driving cell death. Proteases are enzymes that catalyze the breakdown of proteins. They play an essential role in programmed cell death. They're also involved in other cellular remodeling events such as stem cell fate determination, dendrite trimming, inflammation, spermatogenesis, and cell differentiation. Thousands of potential caspase substrates have now been identified. You can see a table of the caspases here for apoptosis and pyroptosis, which is another type of cell death. Here's yet another look at intrinsic versus extrinsic apoptosis, and here you can see the caspases involved with each. Note the death receptor being activated in extrinsic and the mitochondrial permeability that's occurring with the formation of the apoptosome in intrinsic. Apoptosis evolved early in animal evolution and is essential for the development and maintenance of all animals. It is part of the development of hands and feet in humans and paws in other mammals. Interference of apoptosis may contribute to some cancers. That concludes cellular response. We've gone through all three stages of cell signaling: signal reception, signal transduction, and cellular response. As an exercise to tie it all together, let's look at the signaling pathways of cellular responses: metabolism, growth, gene expression, and apoptosis. Response number 1, metabolism. Glucagon or adrenaline are activating the breakdown of glycogen to produce more glucose for energy. These signals bind to GPCR, which activates adenylyl cyclase to cleave ATP and form cyclic AMP. CAMP is the second messenger, which activates PKA, protein kinase A. This begins a phosphorylation cascade, ultimately resulting in the breakdown of glycogen into its glucose components. Glucose can then enter the bloodstream to create more energy in the form of ATP. This is a cytoplasmic event rather than a nuclear event. Response number 2, growth. Here, epidermal growth factor, EGF, binds its receptor, which is a tyrosine kinase receptor. Dimerization of the RTK and autophosphorylation occurs. This activates a relay protein, which triggers a phosphorylation signal cascade. The result is twofold. Some proteins enter the nucleus to affect gene expression. Others remain in the cytoplasm to stimulate translation of proteins. RAS is an important aspect of this pathway. And just as an aside, the gene encoding RAS is an oncogene that was originally discovered when mutations in the RAS protein were linked to cancer. Further studies have indicated that 30% of cancer cells have a mutation in the RAS gene that leads to uncontrolled growth. If left unchecked, uncontrolled cell division can lead to tumor formation and metastasis, the growth of cancer cells in new locations of the body. Response number 3, gene expression. If you look inside the nucleus of this diagrammed cell, you can see all the pathways that are inducing gene regulation and cell proliferation. Gene regulation is generally expressing or blocking the expression of a particular protein. We can use this same diagram to look at apoptosis in the bottom left corner. Here we see both intrinsic and extrinsic apoptosis. Extrinsic is activated by the binding of death factors to the death receptor. inducing caspase 8 activity. Intrinsic is triggered by the cytokines, which induces release of cytochrome C from the mitochondria and caspase activation. And that concludes this lecture. I hope now you can look at this schematic and understand how all the components interact with one another. Again, if you'd like a copy, you can check out the description box below. This is a mind map, which is my absolute favorite way to take notes for biological processes. I hope this was helpful. If it was, please let me know in the comments below, as well as what other lectures you'd find helpful in your studies. Best of luck in your classes. Thank you so much for watching, and I'll see you in the next lecture.